Good afternoon, everyone. I'm Sapna Tripathi, Assistant Professor, IIMT College of Pharmacy. Today, I will discuss about drug absorption, which is a very important topic of biopharmaceutics in B.Pharm third year and M.Pharm first year. So, first, we will discuss about what is absorption. Absorption is the process of a drug moving from its site of delivery into the bloodstream, or you can say the transportation of the unmetabolized drug from the site of administration to the body's circulation system. So we are saying transportation. Why the transportation of drug is required? In order to reach its receptor and produces biological function, a drug should be transported. Now, the drug that have to enter the systemic circulation to exert their effect can be administered by three major routes. The first one, the internal route, and the second one is the parenteral route, and the third one is the topical route. First, the internal route. It includes gastrointestinal, sublingual, buccal, and rectal routes. The gastrointestinal route is the most common for administration of majority of the drugs. The second one is the parenteral route. It includes all routes of administration through or under one or more layer of skin. While no absorption is required when the drug is administered intravenous, it is necessary for extravascular parenteral routes like the subcutaneous and the intramuscular routes. The third one is the topical route. It includes skin, eyes, or other specific membranes. The intranasal, inhalation, intravaginal, and transdermal routes may be considered internal or topical according to their definitions. Now, we can classify these routes. The first one is the oral route, which is the most common route. Approximately 90% of the drugs given by oral routes like tablets, capsules, syrups, and suspension. Second one is the buccal route. Buccal route is when the medicament is placed between the cheeks and the gums, and the sublingual route is the route when the drug is placed under the tongue. Rectal route is used like the suppositories. Suppositories used by the rectal route for local action. Next one is the transdermal route. Transdermal route is the topical route when the medicament is applied on the skin. And this is the slowest absorption route. Next one is the intramuscular route. Intramuscular routes when the drug is given IM into the arms or the thighs or in the buttocks. That is the intramuscular route. Next is the subcutaneous route. Subcutaneous route, as you know, subcutaneous is the internal layer of skin, the dermis, epidermis, and the subcutaneous. In the subcutaneous route, the implants are used for prolonged effect. And next one is the inhalation route. For inhalation route, the volatile drugs are used like for asthma. Next one in intranasal route. Intranasal route is used for the local action. And next one is the intraocular route. Intraocular is the eyes. It is also for the local action. And the last one is the vaginal route. It is used by the vaginal routes. The vaginal tablets or the creams are used for infections. Gastrointestinal absorption of drugs. The oral route of drug administration is the most common for systemically acting drugs, and therefore more emphasis will be given to the gastrointestinal absorption of drug. Moreover, it covers all the aspects of variability observed in drug absorption. Next, the mechanism of drug absorption. There are the 5 mechanisms of drug absorption. First is the passive diffusion or non-ionic diffusion. Second one is pore transport, convective transport, or paracellular transport. Third one is the carrier mediated transport. Fourth one is the ion pair transport, and the last one is endocytosis. First, we talk about passive diffusion. It is also called non-ionic diffusion. This is the major process for absorption of more than 90% of the drug. The driving force for this process is the concentration gradient and no energy required for this process. Passive diffusion is best expressed by Fick's first law of diffusion. Fick's first law of diffusion states the drug molecules diffuses from a region of higher concentration to the lower concentration until equilibrium is attained, and the rate of diffusion is directly proportional to the concentration gradient across the membrane. This is the Fick's equation. dq by dt equal to dak by h multiplied by cgit minus c, where dq by dt is the rate of drug diffusion, d is the diffusion coefficient, a is the surface area of absorbing membrane, k is the partition coefficient, h is the thickness of the membrane, and cgit minus c is the concentration gradient, or you can say difference in concentration of drug in GI fluid and the plasma. The second mechanism is pore transport. It is also called as convective transport, bulk flow transport, or filtration transport. This mechanism is responsible for transport of molecules into the cell through the protein channels present in the cell membrane. There are some characteristics of pore transport. The first, the driving force is constituted by the hydrostatic pressure or the osmotic differences across the membrane, due to which bulk flow of water along with small solid molecules occurs through such aqueous channels. Water flux that promotes such a transport is called solvent drag. The process is important in the absorption of low molecular weight, less than 100 Dalton, and low molecular size, smaller than the diameter of the pores or the protein channel, and generally water-soluble drugs through narrow aqueous-filled channels or pores in the membrane structure, for example, urea, water, and sugar. Chain-like or linear compounds of molecular weight up to 400 Dalton can be absorbed by this mechanism. This is the diagram of pore transport. There is a protein channel in plasma membrane by which the drug is transported from extracellular fluid to the intracellular fluid. Third mechanism is the carrier-mediated transport. It is also called facilitated diffusion. It is a passive diffusion, but because of the presence of some physiological barriers, a carrier is required for the transport. Carrier can be any enzyme, protein, or any other molecule. Binding of drug with the carrier can be reverse or irreverse. There are some important characteristics of carrier-mediated transport. First, a carrier protein always has an uncharged outer surface, which allow it to soluble within the lipid of the membrane. Second, the carrier have no directionally. They work with same efficiency in both directions. The transport process is structure specific. The carriers have special affinity for and transfer a drug of specific chemical structure. Only generally the carriers have special affinity for essential nutrients. As the number of carrier is limited, the transport system is subjected to competition between agents having similar structures. This is the diagram of facilitated diffusion or carrier-mediated diffusion. See, there is vitamin B12 in gastrointestinal lumen. It requires a carrier to cross this membrane. When it attached to a carrier, it easily crosses the membrane and reaches to the blood. When it reaches to the blood, it dissociated. The fourth mechanism is ion pair transport. Yet another mechanism that explains the absorption of drugs like quaternary ammonium compounds and sulfonic acid which ionize under all pH conditions, is ion pair transport. Despite their low oil in water partition coefficient values, such agents penetrate the membrane by forming reversible neutral complexes with endogenous ions of the GIT like mucin. Such neutral complexes have both the required lipophilicity as well as aqueous solubility for passive diffusion. Such a phenomena is called ion pair transport. Propranolol, a basic drug that forms an ion pair with oleic acid, is absorbed by this mechanism. And this is the diagram of ion pair transport. See, in GI lumen, a cationic drug forms a neutral complex with the endogenous ion. Then it easily crosses membrane and reaches to the blood, and when it reaches to the blood, it dissociated. And the last mechanism is endocytosis. It works on the principle like the amoeba engulf their food. It is a minor transport mechanism which involves engulfing extracellular material within a segment of cell membrane to form a saccule or a vesicle, which is then pinched off intracellularly. This is the only transport mechanism whereby a drug or compound does not have to be in an aqueous solution in order to be absorbed. This is the endocytosis diagram. Endocytosis can be of two types, the phagocytosis and the pinocytosis. Phagocytosis is the cell eating where the solute particle is a solid, and pinocytosis is cell drinking where the solute particle is in the liquid form. Next, when we talk about the factors affecting absorption of drug, there are many factors, but we can classify them in the two categories. First one is the pharmaceutical factor and the second one is the patient-related factor. Pharmaceutical factor include factors related to the physicochemical properties of drug and dosage form characteristics and pharmaceutical ingredients. We can further classify pharmaceutical factors into the two categories. First one is the physicochemical properties of drug substances. It includes drug solubility and dissolution rate. You know that the higher the drug solubility, higher will be the absorption rate. Second one is the particle size and effective surface area. Particle size is inversely proportional to the absorption of drug, means higher the particle size, lower is the absorption. So particle size should be minimum for better absorption. Next one is the polymorphism and amorphism. Next, the pseudo-polymorphism. Next, the salt form of the drug. Next, the lipophilicity of the drug. As you know, for the better absorption of the drug should be lipophilic. Next, the pK of the drug and gastrointestinal pH. Next one is the drug stability, and the last one is stereochemical nature of the drug. The pharmaceutical factor is further divided in second category, which is dosage form characteristic, or you can say pharmacotechnical factors. The first one is the disintegration time. It is generally for tablets and capsules. Second one is dissolution time. Third is the manufacturing variables. Fourth one is the pharmaceutical ingredients or the excipient or additives which are used for the manufacturing. Fifth one is the nature and type of dosage form. And sixth one is the product age and storage condition. These all factors affect the absorption of the drug, which like disintegration time. And disintegration time should be less for better absorption. Same with the dissolution. Less dissolution time required for better absorption. The second factor is patient-related factor. It includes factors relating to the anatomical, physiological, and pathological characteristic of the patient. The first one is the age. As you know, in the children's and in the old patient, the absorption of drug is slower in comparison to the adults. Same with the gastric emptying time. Higher will be the gastric emptying time for better absorption of drug. Next, the intestinal transit time. For better absorption of drug in intestine, the transit time should be higher. Next, the gastrointestinal pH. Fifth one is the disease state. As you know, when the person is suffering from disease, any diseases, the metabolism and absorption of person is become slow. And the last one is blood flow through the GIT. These are the factors which affects absorption of the drug. Thank you. That's all for today.